At the heart of every living thing lies DNA, the blueprint of life, dictating everything from eye color to disease risk. For centuries we could only read this code, powerless to change it. But what if we could edit it? Imagine correcting a single typo in a 3 billion letter manual. Molecular editing now lets us do just that, precisely changing DNA or RNA at will. This power could eradicate genetic diseases like cystic fibrosis and sickle cell anemia by fixing their root causes. For generations, medicine treated symptoms. Now we can target the source. As we enter this new era, understanding gene editing is crucial for everyone, not just scientists. This journey will explore the fundamentals, applications, challenges, and future of molecular editing. Molecular editing is a groundbreaking set of technologies that allows scientists to make incredibly precise changes to DNA or RNA. Think of it as a genetic find and replace function, but at the molecular level. This means we can now target specific sequences in our genetic code and alter them with a level of accuracy that was once unimaginable. Unlike older forms of genetic engineering, which were often imprecise and could cause unintended changes, these new tools act like a scalpel guided by GPS, allowing for targeted, safer, and more predictable edits. This precision greatly reduces the risk of unwanted side effects. And editing isn't limited to DNA. Scientists can also modify RNA, the molecule that helps turn genetic instructions into action. RNA editing offers temporary reversible fixes for certain conditions, making it a flexible option for treating diseases or studying gene function. This empowers researchers to explore how genes work, enables doctors to treat diseases that were once incurable, and helps agricultural scientists develop crops that are more resilient, nutritious, and sustainable for the future. Each tool in the molecular editing toolbox, like CRISPR, TALEN, or zinc finger nucleases, is designed for specific types of genetic modifications, giving scientists a wide range of options for different challenges. The result? We now have unprecedented control over life's fundamental building blocks, opening up possibilities that were once the stuff of science fiction. With these tools, we can turn genes on or off, insert entirely new ones, or correct harmful errors with remarkable accuracy, paving the way for new therapies and discoveries. This technology is revolutionizing medicine, transforming agriculture, and accelerating basic research, promising a future where genetic diseases can be treated at their source and food security is improved worldwide. Molecular editing is not just a tool, it's a new way to interact with life itself, giving us the power to shape the future of biology and human health. The most famous and revolutionary molecular editing tool in modern science is CRISPR-Cas9, a technology inspired by a remarkable bacterial defense system that protects microbes from invading viruses. Bacteria use CRISPR to capture and store snippets of viral DNA, creating a genetic most wanted list that helps them recognize and fight off future attacks. The CRISPR system is made up of two main components: a guide RNA, which acts like a molecular bloodhound, searching for the matching DNA sequence, and Cas9, a specialized protein that functions as precise molecular scissors, ready to cut the DNA at the right spot. When the guide RNA finds its perfect match, Cas9 is activated and slices the DNA at that exact location, creating a targeted break. Scientists soon realized they could reprogram this system, designing custom guide RNAs to target virtually any gene in any organism, from bacteria to plants and animals. After the DNA is cut, the cell's natural repair machinery takes over, sometimes introducing changes or corrections that scientists have supplied, or simply patching the break. This powerful method allows us to disable faulty genes, correct mutations, or insert beneficial new genes with incredible precision and control. CRISPR-Cas9 has made gene editing not only faster and more efficient, but also dramatically cheaper and more accessible to researchers around the world. It's a scientific breakthrough that lets us rewrite the genetic code with a level of accuracy and ease that was once unimaginable. The possibilities are vast and exciting, from curing genetic diseases, to engineering crops with better yields, to creating entirely new traits in living things. CRISPR has truly democratized genetic editing, opening the door to a future where anyone, anywhere, can help shape the code of life. Molecular editing is already transforming medicine, agriculture, and research, opening up possibilities that were once the stuff of science fiction. With each breakthrough, we're learning to rewrite the very code of life, unlocking new ways to solve some of humanity's biggest challenges. In medicine, CRISPR is being used in clinical trials to cure genetic diseases like sickle cell anemia by correcting faulty genes in a patient's own cells. This technology offers hope to millions suffering from inherited disorders and could one day eliminate these conditions entirely. It's also being explored to engineer immune cells to fight cancer more effectively, and even to target viruses like HIV, potentially leading to new treatments and cures for diseases that have long eluded us. In agriculture, CRISPR enables the creation of crops that resist drought, pests, and disease, and even improves nutrition without introducing foreign genes. Farmers can grow more resilient plants, helping to feed a growing global population. Researchers use gene editing to uncover how genes work, accelerating discoveries in biology and paving the way for new therapies and innovations. These advances promise healthier lives, more resilient food supplies, and deeper scientific understanding, benefiting people and communities everywhere. The impact is global, touching everything from our dinner plates to the frontiers of medicine, and connecting scientists and doctors across continents in a shared mission. CRISPR is reshaping our world, one gene at a time, and its story is only just beginning. With great power comes great responsibility. Editing genes raises profound ethical questions. Somatic editing, which changes only an individual's body cells, is widely accepted for treating disease. Germline editing, which alters genes passed to future generations, is far more controversial. It could eliminate hereditary diseases, but the risks and unknowns are enormous. There's also concern about designer babies and genetic inequality, where enhancements could deepen social divides. Who decides what traits to edit, and who gets access? These are questions society must answer together. As gene editing advances, global dialogue and clear regulations are essential to ensure it benefits all humanity. The future of molecular editing is rapidly evolving. New tools like base and prime editors allow even more precise, safer changes, like using a pencil instead of scissors. Soon, gene editing could become routine in medicine, preventing diseases before they start and even targeting aging itself. In conservation, we could engineer species to survive climate change or restore genetic diversity. Agriculture may see crops designed for sustainability, reducing the need for chemicals. The synergy with AI and synthetic biology could let us design new life forms to solve global challenges. We're only beginning to glimpse the possibilities of this new scientific era. We now hold the power to rewrite life's code, a responsibility as immense as the opportunity. Molecular editing offers hope for curing diseases and building a sustainable future, but it also demands ethical wisdom. The choices we make today will shape generations to come. We must balance scientific progress with caution, ensuring robust global norms and equitable access. This technology is a mirror of our hopes and fears, challenging us to define what kind of future we want. Will we use gene editing to heal and uplift, or to divide and exclude? The story of molecular editing is ours to write, together. By embracing both science and ethics, we can steer this power toward a healthier, more just world.